Here is the graph of the membrane potential of a neuron. Action potential is a moving electrical impulse triggered by a change in charge distribution. Resting potential is the difference in charge across the membrane. It contains the potential energy needed to transmit an impulse. The resting state is when the gated sodium and potassium channels are closed. Depolarization occurs when sodium travels down its concentration gradient through ion-gated channels. Depolarization opens up most of the sodium channels, but the potassium channels stay closed. Next, the sodium channels become inactivated, but the potassium channels open up. Finally, undershoot or hyperpolarization is when the membrane potential becomes more negative. It is the opposite of depolarization. It happens when sodium channels close, but some potassium channels are still open. This is when the membrane returns back to its resting state. Voltage-gated ion channels open and close based on the membrane potential and the concentration of sodium and potassium ions.
In the presynaptic terminals, synaptic vesicles store various neurotransmitters that are released into the synaptic cleft, the area between pre- and postsynaptic neurons at chemical synapses. Action potentials activate sodium and potassium channels. Those end up activating voltage-gated calcium channels, which cause vesicles to fuse with the cell membrane and send out the neurotransmitters. The saltatory conduction is a type of nerve impulse that allows signals to pass much faster in myelinated neurons. The signal jumps from node to node because action potentials propagate down the axon. At each node, the membrane is depolarized and the action potential is regenerated. There are two types of neurotransmitters that interact with receptor proteins on the postsynaptic membrane: inhibitory and excitatory. Excitatory neurotransmitters stimulate the brain. They depolarize the postsynaptic neuron by making the inside more positive and bringing it closer to its action potential threshold. Inhibitory neurotransmitters hyperpolarize the postsynaptic neuron by making the inside more negative, driving the charge away from the threshold.